Hi everyone. I will be presenting my research on plasma phospholipid fatty acids in acute myocardial infarction, a Mendelian randomization study. Acute myocardial infarction, or AMI, is a severe condition that blocks blood flow to the heart muscle due to blocked arteries and bears a high mortality rate among cardiac patients. It is also commonly referred to as a heart attack. However, a suboptimal diet has been suggested as an essential AMI risk factor that could be prevented through the dietary intake of polyunsaturated fatty acids, or PUFAs. Here in Figure 1, you will see the synthesis of omega-3 and omega-6 family of fatty acids. The supplementation of these PUFAs have been suggested by previous studies to affect the incidence of AMI. But many recent studies have inconsistent results. Therefore, this proposal aims to determine the causal association between specific PUFAs and the incidence of AMI. This can be done by using the Mendelian randomization approach, which is best explained by this flowchart shown in Figure 2. An instrumental variable or genetic variant called single nucleotide polymorphisms, or SNPs, are used to determine the causal association between an exposure or non-genetic risk factor, in this case being the PUFAs, and an outcome or disease, in this case being AMI. Through this process, unmeasured confounding factors are minimized because genetic variants are randomly assorted during meiosis or conception. A two-sample MR design was used to determine the causality between the PUFAs and AMI. First off, using previously conducted meta-analysis of genome-wide association studies from the CHARGE consortium, 16 nucleotide polymorphisms, or SNPs, were selected as instrumental variables. The SNP fatty acid association were at genome-wide significance level, or 5 times 10 to the negative 8, as shown here in Table 1. More details can also be found here. The outcome data was retrieved from the UK Biobank, with a total of 7,045 AMI cases found after quality control. The SNP AMI association was found from 310,999 participants from the UK Biobank after quality controlling for these 7 factors here. Then, the genome-wide association study was conducted to find the causal association between the SNP chosen as instrumental variables and the incidence of AMI. Finally, the statistical analysis conducted was the traditionally used inverse variance weighted method, or IVW, and the Walt ratio. In Table 2, you can see the results from the IVW method and the Walt ratio. The Walt ratio is used for plasma PUFAs with only one SNP fatty acid association, whereas the IVW is used for more than one. As you can see, none of the p-values appear to be significant. Also, Figure 3 is a visual representation of the odds ratio plot that more clearly shows that there is no significant association between the PUFAs and the incidence of AMI. It also has a p-value right here. This may be indicative that although the dietary intake of polyunsaturated fatty acids has been previously associated with acute myocardial infarction, it is not a causal association according to the results. However, the results need to be confirmed by performing this study while considering weak instrument bias. Therefore, it is recommended that similar studies use the generalized summary database Mendelian randomization method in the future. Thank you.